Thank you for participating in the Noble Conference. My name is Dr. Stephen Fruchtman. I'm the president and CEO of Onconova Therapeutics. I'm a hematologist, oncologist by training. I had an academic career running the bone marrow stem cell transplant program at Mount Sinai in New York for 20 years. I was the clinical chief of hematology as well. And I've been blessed to have gotten a number of drugs approved by the US FDA for patients with advanced hematological and solid tumors. And our mission at Onconova is to continue to develop new drugs for patients with advanced cancers. I will be making forward-looking statements as is customary. So Onconova is a clinical stage biopharmaceutical company. As I mentioned, we are focused on developing novel drugs to study in patients with advanced cancer to hope to improve upon the therapeutic options for them. Our drugs are developed internally and with the aim of disrupting key pathways in how cancer cells both metastasize and proliferate. Our key compound now in early development is ON-123-300. I will tell you all about the mechanism of action and what tumors we plan to target with ON-123-300. We are also developing oral Regor-sertum in combination with an immuno-oncology agent, nivolumab, in KRAS-mutated cancers. So some of our financial highlights are listed on this slide. End of September, we had $24.2 million in cash. We recently raised $8.7 million in early January with institutional investors, some key biotech investors. So we have in the high 20 millions, which will be sufficient to develop ON-123-300 through the phase 1 study and including an expansion study when we plan to target specific indications that look fruitful for the patients who have those diseases. And we're always looking to in-license potential opportunities, as I mentioned, that we have developed over my career and along with my colleagues, key drugs in hematology oncology, and would like to continue to do that. So what is ON-123-300? It's a third generation multi-kinase inhibitor. That means it attacks multiple kinase pathways involved in cancer, and most specifically targets CDK4 and 6 and ARK5. I'll take you through the science of that shortly. CDK4/6 are approved by the US FDA. There are 3 such drugs that target CDK4 and 6. They are only approved in hormone receptor positive, meaning the breast cancer cell has receptors for either estrogen, progesterone, or both. These cancer cells are HER2 negative, and they have already metastasized outside the breast. They are always given in combination with an aromatase inhibitor, and I'll take you through the thinking of why that is. We have already opened a phase 1 trial with ON-123-300 in China. Our IND for the US study was approved late 2020, and we anticipate first patient entering the phase 1 trial some point in the first half of 21. Regor-sertum is being developed in KRAS-driven cancers in multiple investigator-sponsored combination studies with immuno-oncology drugs, and the lead indication currently is in KRAS advanced mutated non-small cell lung cancer. We are committed to new business development, both to in-license compounds, as I mentioned, to expand our product pipeline, and we have the ability to out-license to certain geographies our current pipeline products. So what is ON-123300? This is a pictorial of the cyclin D4/6 pathway in cancer. And this complex slide shows us how in the nucleus, cyclin 4 and 6 turn on pathways that change the cell status from a quiescent cell in the G1 phase into a synthetic cell, meaning once cyclin 4 and 6 are overexpressed, cancer cells then enter a synthetic pathway where they replicate, they copy their DNA, they replicate and they proliferate. And that's how cancer cells have a proliferative advantage over normal non-cancerous cells. One of the mechanisms is by overexpression in cancer cells of cyclin D4 and 6. And there are 3 drugs listed here that target the overexpression of CDK4 and 6. The 3 drugs are palbociclib, ribociclib, and abemaciclib. And these are multi-billion-dollar franchises for the big pharma companies that produce these drugs and are used in women, unfortunately, who have metastatic breast cancer. I will mention that none of these drugs are curative. All the women will progress. But here is an outline of the CDK4/6 marketed products. As you can see, the first drug approved for this indication was in 2015, Ibrance, and you could see how the other drugs got subsequent approval and how these franchises are growing to be multi-billion-dollar assets. Of course, they do have some potential serious adverse events as listed here. I want to highlight to you that palbociclib, the most commonly prescribed CDK4/6 inhibitor, causes neutropenia. Some of the women need to get growth factor support to prevent that from happening. Ribociclib causes QT prolongation and abnormal EKG, which can be associated with sudden death. And abemaciclib causes severe diarrhea. All of these drugs, because they are not cytotoxic, have to be given in combination with an aromatase inhibitor. So they cannot be used as single agents. Here is the biochemical structure of ON-123300 compared to Ibrance or palbociclib. You can see there was minor changes made in the chemical structure of the drug in order to attack other kinases overexpressed in cancer. Specifically, as you see the inhibitory concentrations listed here, you can see how CDK4 and D1 inhibition are very similar between ON-123300 and Ibrance. However, the target ARC5 has 4.95 nanomolar concentration in order to inhibit 50% of the activity, while palbociclib requires over 5,000 nanomolars to inhibit 50% of the activity. In addition, ON-123300 targets other pathways very important in cancer. And the comparison IC50s, the inhibitory concentration, 50%, again, are listed here. In addition to CDK4, 6, and ONC5, you can see why ON123 is a multi-kinase inhibitor. I will point out to you the FLT3 pathway. About 20 to 30% of patients with AML have mutations of FLT3. There are 2 drugs already approved by the US FDA for FLT3 mutated acute myeloid leukemia. Right now, we are planning to focus on solid tumors, but clearly we'll continue to investigate hematological malignancies as well, and one day perhaps FLT3 mutated AML. So the potential, based on the mechanism of action of this drug, is very diverse. I already mentioned that the commercially approved CDK4/6 inhibitors have to be given in combination with the second drug, an aromatase inhibitor. The reason for that is the approved drugs are what's called cytostatic, meaning it prevents cancer proliferation, but it does not kill the cancer cell. ON123 as I'll show you in a subsequent slide, is actually cytotoxic. Thus, there is a potential that these patients can be treated with a single agent. The doublet, of course, has side effects associated with it, making it, in some patients, difficult to tolerate. Aromatase inhibitors are estrogen blockers, and some of the patients have difficulty with estrogen blockade. If we can prove that ON123 can be given as a single agent, if it's efficacious for that indication, the issue of tolerability may be reduced. In addition, ONC5, in addition to metastatic breast cancer, is overexpressed in other diseases as well, including colorectal cancer, multiple myeloma, and hepatocellular carcinoma. I will show you our preclinical data for a number of these indications. So, how does cancer kill patients? I will highlight breast cancer because that's where the CDK4/6 inhibitors are already approved. So women with localized breast cancer to their breast do not die typically of breast cancer. They can be treated with surgery, with radiation. The cause of death is metastatic disease. For a breast cancer cell to metastasize, it has to leave its natural environment in the breast. It is not well understood how that occurs, but it is believed, and there is data suggesting that the ONC5 pathway is involved in the process as delineated here. Once the cancer cell detaches from the breast tissue, it has to migrate into a blood or lymphatic vessel. It has to invade and get through the vessel wall to get into the vessel, and then travel through the blood or lymphatic supply to every cell of our body. Why it attaches to some environments and not others is, again, not well understood. But to attach, let's say, to the liver, to metastasize to the liver, again, it has to reattach itself to the vessel wall feeding that liver. It has to extravasate outside of the blood vessel wall into the liver, proliferate, and have a proliferative advantage over the normal liver cells. It is this metastatic process that ultimately causes the demise of many patients with solid tumors, and breast cancer can metastasize to the lung, liver, the brain, and the skin as well. So anything that can be done to prevent metastatic disease or minimize it would be beneficial for patient care. So here is a pictorial of how ONC5 works. I will point out the photomicrograph number B. You can see these are 2 mice. The PanC1 mouse is a mouse with implanted human pancreatic cancer. You can see the nodule on the hind limb of the mouse. P/ARC, the mouse underneath that, again, is a pancreatic cell line implanted into a mouse, but this pancreatic cell line has overexpression of the ARC5 pathway. And you can see the profound difference in the size of the tumor between these two mice, and those size differences are depicted in photomicrograph A, the bar graph. You can see when you measure that pancreatic cancer in the mouse, the one that overexpresses ARC5 is 10 times, approximately 10 times the size. Photomicrograph C shows, again, the PAN-C1 mouse looking at that tumor under a microscope, and the large area N represents where the tumor is necrotic. That's why it's the letter N. Note under ARC5 overexpression, there's much less necrosis. There is some necrosis, but much less necrosis when ARC5. So these tumors can grow larger because there's fewer cancer cells that are dying. This is another study comparing ON123 to palbociclib. The number for palbociclib is PD0332991. That's palbociclib or Ibrance. This is a wound healing model. So for a wound to heal, cells have to migrate. So wound healing models are used to understand how cells metastasize and invade tissues. As you can see, there is much that ON123 is much more efficient at inhibiting wound healing. In other words, if the wound healing is inhibited, it's because cells cannot migrate to that wound and promote healing. For a wound to heal, cells have to migrate to that wound and promote healing. If those cells can't get there in a wound model, it can't heal. Well, in metastatic disease, clearly you don't want the breast cancer cell to get into another environment which can cause metastasis. So this wound healing model is used to study metastasis, and ON123 prevents wound healing and hopefully will prevent metastatic disease as well. I mentioned that palbociclib, one of the side effects is neutropenia. This is a comparison in a preclinical model of palbociclib versus ON123, looking at neutrophil counts, the bar graph to the right. As you can see in the gray bar, the control, what the white count is. You can see palbociclib decreases that white count significantly, but ON123 decreases the neutrophil count less. We hope when we put this ON123 compound into the human model, we can replicate this preclinical data. If ON123 causes less neutropenia, it will be a major clinical benefit for patients receiving our drug. So to summarize and differentiate ourselves from the FDA-approved CDK4/6 inhibitors, we have the potential, because we are cytotoxic, we kill cancer cells, we don't just prevent their proliferation, to be used as a single agent. I will show you data where ON123 is active in CDK4/6 resistant tumor models. Because we have a broader spectrum of kinases that we inhibit, we have the possibility for broader indications, tumor indications, other than metastatic breast cancer. And we will be hopefully one day studying these additional indications. So this is the data I was alluding to, comparing ON123 to palbociclib, again, PD0332991. You can see in breast cancer cells resistant to palbociclib in the red line how ON123 controls tumor proliferation. That is true for prostate cancer cells and ovarian cancer cells as well. So cancers resistant to palbociclib are sensitive to ON-123300 in these preclinical models. This is another disease, mantle cell lymphoma, engrafted again into a mouse. You can see in photomicrograph B how the control-treated mouse has a massive mantle cell tumor on its right hind limb. And look what happens to the mouse treated with ON-123300. The mantle cell volume is greatly controlled. Here is a listing of the type of diseases we plan to study one day. Clearly, we will study in the phase 1 all advanced cancers to establish the recommended phase 2 dose, but we hope to include some women in the phase 1 who have failed the CDK4/6 approved drugs and have agreed to participate in our phase 1. But as part of the expansion, we clearly will focus on women with metastatic breast cancer, hormone receptor positive, HER2 negative, who have progressed on the CDK4/6 approved drugs, so we can study the potential efficacy, and hopefully the preclinical models will successfully be translated into a clinical model. Other diseases as listed here also have the possibility of being studied because CDK4/6 is overexpressed as well as ARC5. So we filed the IND at end of 2020. We have approval. We anticipate putting the first patient into the phase 1 in the first half of 2021. We already have a phase 1 study up and running in China. These studies are a bit different. The study in China treats the patients 3 out of 4 weeks. The study in the US is continuous dosing. The reason for those differences is because the FDA-approved products in the US, the different products are given in a different manner. Some given 3 out of 4 weeks, some given continuously. So we are recreating those models. That is the presentation for ON-123300. We'll hold questions for the end, but I'd like to move on now to the development of oral Regor-sertum in KRAS mutated cancers. We have already opened a phase 1 investigator-initiated study at Mount Sinai Hospital in New York. We have already gone into 3 cohorts. This is a combination approach in patients with KRAS mutated non-small cell lung cancer who have already progressed on the standard of care, which typically includes an immuno-oncology approach. So patients with advanced KRAS mutated non-small cell lung cancer are entered onto this trial in collaboration with Bristol Myers Squibb, who've been kind enough to provide free nivolumab. So all the patients get free and full dose nivolumab, and we gradually increase the dosing of Regor-sertum. We're already at 560 milligrams of oral Regor-sertum twice a day. To date, this is the highest oral drug we've given, but the combination to date is well tolerated. And we anticipate once we determine the optimal combination study to continue to study in a phase 2 manner this combination in a variety of KRAS mutated cancers using the investigator-initiated approach. So how does Regor-sertum work? One of the mechanisms is to negatively impact on the mutated RAS pathway. So RAS is the most common mutation in cancer. There are different types of RAS mutations. The patient here had a KRAS mutation involving the site G12D. It was metastatic, and it was put into a patient-derived xenograft mouse model. As you can see, when placebo is used to treat the mouse, the tumor continues to grow in the blue line. Regor-sertum in the orange line controls the tumor growth. The photomicrographs below are very interesting. So RAS is the mutated gene making an abnormal protein, giving the cancer cell a proliferative advantage, and that RAS mutated pathway ends in ERK, in p-ERK. The upper photomicrograph, in 5 different mice, you can see the brown material. That is staining of ERK. So you can see when placebo is given, there's a lot of ERK present in the tumor. Notice when Regor-sertum in the lower photomicrograph, again, 5 different mice, KRAS mutated lung cancer. When Regor-sertum is given, there's much less brown staining material because the mutated RAS to p-ERK pathway has been downregulated and hopeful to control the tumor. The other thing Regor-sertum does, I've mentioned immuno-oncology, a very exciting area that has revolutionized cancer therapeutics for a number of indications. But for immuno-oncology drugs to work, the tumor has to be infiltrated by lymphocytes. That the lymphocytes recognize the tumor as causing harm. The immuno-oncology drugs stimulate those lymphocytes to attack the tumor and hopefully to control it. But for that to occur, there has to be sufficient lymphocytes infiltrating the tumors. Not all tumors have that. Tumors that do not have sufficient numbers of lymphocytes surrounding the tumor are called cold tumors or cold lymph nodes. Regor-sertum has the ability to attract lymphocytes to target the tumor and to transform a cold tumor or a cold lymph node draining the tumor into a hot lymph node or a hot tumor because of the presence of more lymphocytes as highlighted by CD3 staining, and thus to give the immuno-oncology drugs a chance to work and control the tumor. And this formed the foundation of the trial at Mount Sinai I already showed you. In addition, it's not just non-small cell lung cancer. KRAS is commonly mutated in colorectal cancer as well. As you see in the upper left-hand corner, this is a colorectal cancer model. Again, placebo versus Regor-sertum in the blue line with the pictures of the mice below. And you can see how Regor-sertum controls tumor growth. Non-small cell lung cancer, again, similar story. Placebo given to the mice, the tumor grows. In the presence of Regor-sertum, tumor control is achieved. Finally, this is a combination approach in colorectal cancer of Regor-sertum and a PD-1 inhibitor. In this colorectal cancer model, when placebo is given in the upper blue line, you see how the tumor grows. Regor-sertum in the orange line controls the tumor growth. A PD-1 inhibitor in the purple line controls the tumor as well. But look at the green line. Look at the synergy when Regor-sertum and PD-1 inhibition is given together, how profoundly that tumor growth is controlled. And again, these are already in human models. We're studying the combination of Regor-sertum and a PD-1 inhibitor such as nivolumab. So summary and upcoming milestones. Here's our cash position, which I already mentioned. We have in the high $20 million range, which is sufficient to fund the studies I have mentioned to you and develop ON123-300 through a phase 1, including the expansions part of that phase 1, and looking at potential in-licensing opportunities as well. Regor-sertum is already and will be in multiple investigator-sponsored trials in KRAS mutated solid tumors. We are committed to new business development. We have the opportunity, we hope, and are actively looking for in-licensing compounds to expand our product pipeline. from ON123-300 and oral Regor-sertum to include other opportunities, and we can out-license our products to various geographic areas as well. Phase 1 for ON123-300 already is open in China. 2 cohorts are open. The initial dose was 40 milligrams. Next dose was 80 milligrams. That's where we're at now, and to date, no dose-limiting toxicities have been encountered. Anticipate opening a trial in the U.S. first half of 21. I believe I've mentioned all of these things to you already, and I appreciate your time today and hope this talk was clear, and be happy to take any questions.
Thank you so much for the presentation, Steve. It was very informative. So I will ask a few questions, especially the CDK4/6 ARC-5 program. My first question would be, so the currently approved drugs targeting CDK4/6 develop resistance. And I believe that one of the strategies is going after PI3K, mTOR combination. Also, ARC-5 is important in this pathway. And you mentioned you will be targeting resistant patient population. I am curious if you would also go after refractory patient population that never responded to CDK4/6, or is it not a target population currently?
No, we will study all advanced cancers, and if we focus on metastatic breast cancer, which I think your question was about, a patient who never responds or responds and progresses, which is the majority of patients, will be included. We will not differentiate. They'll both be welcomed onto the study because they have no other therapeutic options other than chemotherapy, so they'd be welcome onto the study.
That's great. And my follow-up question will be, when should we expect any data from this trial? I know the first patient enrollment is expected in near term. So are we expecting any data readout within 2021, or is it going to be later at time?
So as you know, Aho, you never know in a phase 1 when you're going to encounter at what dose and how many patients dose-limiting toxicities. It could happen at 6 patients, it can happen at 9 patients, it can happen at 30. So there's a little bit of an unknown. But we anticipate, because we have 2 cohorts already open in China with no dose-limiting toxicities, we may have the recommended phase 2 dose of ON123-300 sometimes towards the end of 21.
That sounds great, Steve. Thank you so much again for your participation. Much appreciate your presentation at our conference. Thank you.
Thank you so much, Ahu. And again, thanks to Noble for inviting us to present our data. We appreciate it.